infiltrate cells, communicate, surround Reed-Sternberg cells, and even form so-called rosettes. So they stick to Reed-Sternberg cells and communicate in some way. Importantly, there is a very little evidence of effective immune response in this tumor despite this prominent reactive infiltrate. So if you look more carefully at the microenvironment composition, there are T cells, as I said, eosinophils, macrophages, plasma cells, fibroblasts, but within the T cell compartment, some subpopulation of T cells are overrepresented and some are underrepresented. So, for example, Th2 cells and T cells capable of suppressing efficient immune response, such as Treg cells and IL-10 producing cells, are markedly overrepresented, whereas cytotoxic T cells and Th1 cells, so these are the cells that are actually effective in mounting immune response against the tumor, are underrepresented. So this suggests that tumor cells in this disease in some way evade immune attack from the host. So what are the mechanisms responsible for this observation, and can we bypass them, or can we modulate these mechanisms? So to answer these questions, over a decade ago, we started our studies with very simple experiment. We basically compared gene expression profiles between a panel of diffuse large B-cell lymphoma cell lines, which is basically a different type of B-cell malignancy, with this specific lymphoma subtype, Hodgkin lymphoma. And we were specifically looking for molecules that can be involved in immune modulation. And we identified galectin-1, which is an immunomodulatory protein that was markedly overrepresented in Hodgkin lymphoma cell lines. So there was about 25-fold overexpression at the mRNA level in Hodgkin lymphoma cell lines at the mRNA level, but as you can see here at the bottom, at the Western blots, it was also overexpressed at the protein level. There was abundant expression in Hodgkin lymphoma cell lines here in the upper left, whereas in the diffuse large B-cell lymphoma the expression was low or even undetectable. So we looked next at the tissue section. We stained this diagnostic slides with antibodies against galectin-1, and we detected specific overexpression of galectin-1 in Reed-Sternberg cells, in the tumor cells. So the tumor cells, but not the microenvironment, overexpresses galectin-1, and that was not the case in diffuse large B-cell lymphoma, shown here at the bottom. So why the galectin-1 overexpression was important? We thought that it was important because a few years earlier, from our studies, there was a paper published by our collaborator Gabriel Rabinovich, who identified galectin-1 in melanoma. And they showed that in this tumor, overexpression of melanoma is responsible for tumor immune escape. Basically, what they did in this experiment, it's a key experiment in that paper, where they silenced expression of galectin-1 using antisense oligonucleotides, and it was B16 melanoma model, so syngeneic, they transplanted these cells to syngeneic mice and monitored tumor growth after the inoculation. And as you can see here on the right, the tumor cells that had high expression of galectin-1 formed progressively growing tumors, whereas tumors with intermediate or low galectin-1 grew much slower. And that was entirely dependent on T cell presence in the mice, because depletion of the T cells abrogated this effect. So galectin-1 is immunoregulatory protein. And how it basically works, how it exactly works. Galectin-1 is lectin, so it binds glycosylated surface receptor. It binds to N-acetyllactosamine units, and the binding is dependent on terminal sialic acid attached in either alpha-2,3 by alpha-2,3 link, which is permissive to galectin-1 binding, or by alpha-2,6 link, which is not permissive to galectin-1 binding. And it happens so that different subpopulation of T cells express different sialic acid transferases that attach this sialic acid in different configurations. And these T cells that are capable of mounting immune response, so Th1 cells, Th17 cells, and cytotoxic T cells, are permissive to galectin-1 binding, whereas Th2 cells, Treg cells, immunosuppressive cells do not bind galectin-1. And as a consequence, these cells that bound galectin-1 undergo apoptosis and are deleted from the microenvironment, whereas Th2 cells and Treg cells are expanded and overrepresented relatively in this microenvironment. So can we interfere with this mechanism? To answer this question, we developed a panel of antibodies. They happened to exhibit very high affinity to the antigen because they recognize the cognate protein at the 1 to 33,000 dilution. So that was a really potent antibody. And we used that antibody to an in vivo melanoma model. We didn't have Hodgkin lymphoma model, syngeneic model available at the time. We tried to develop it, but we failed miserably in this aspect. So we took advantage of the available model. Basically, it was the B16 melanoma model, syngeneic model. We inoculated 10 million cells to mice, waited until the tumors formed. The cells formed tumors of about 100 cubic millimeters, and they initiated our treatment. And this is the result of this experiment. When we looked at the growth of mice, of the tumors in mice that received only vehicle, as expected, these mice developed progressively growing tumors, whereas mice receiving galectin-1 antibodies, these tumors grew much slower. So to summarize our studies in this tumor and on galectin-1, we found that galectin-1 is uniformly overexpressed in this disease, specifically overexpressed in this disease. It can be also used as a diagnostic marker. It induces apoptosis of activated CD8 T cells and Th-type cytokine production in CD4 cells. And we concluded that galectin-1 expression is a likely basis for the skewed nature of the Hodgkin lymphoma inflammatory infiltrate and the ineffective host anti-tumor immune response. And targeting galectin-1 is a potential immunotherapeutic strategy in these patients. Okay, so after these early experiments in this disease, we wanted to ask a different question. Can we identify structural alterations that would be associated with immune evasion in this disease? And we performed a very similar experiment to that first study comparing gene expression profile. We basically took a panel of diffuse large B-cell lymphoma cell lines, Hodgkin lymphoma cell lines, and performed high-density SNP arrays to identify structural alterations in this disease. And what we encountered here is the very specific amplification of 9p24 locus. And that was fascinating finding because this locus harbors two super important ligands, PDL1 and PDL2, which are known now. Now we know that they are very attractive therapeutic targets. So this amplification was also associated with overexpression of the mRNA level, as you can see here on the right. So why that was important? So what is PDL1 and PDL system? It's a physiologic mechanism to silence immune response at the end of the immune response after antigen clearance. So basically, antigen-presenting cell overexpresses PD ligand, which is then bound by PD1 on the surface of T cells, and this blocks signal transduction activating T cells. And this mechanism is hijacked by tumor cells, which can overexpress PD ligands. And most importantly, this is fully reversible, because if you disrupt this interaction between PDL and PD ligand with PD1 receptor, then you can fully restore the activity of T cells and you can reactivate, reinvigorate them to attack the tumor. So we looked back at our panel of cell lines and we compared the surface expression of PDL1 and PDL2, and we found that they were in proportion to the copy number gain. So the higher was the copy number gain indicated here in parentheses below the cell name, the higher was the expression of the ligand on the surface. So that seemed to be a specific mechanism responsible for overexpression of these ligands. When we looked more closely at this locus, it turned that it harbors not only PDL1 and PDL2, but also JAK2 kinase, which is very well known oncogene in many tumors. And in this aspect, it was particularly important because JAK2 is involved in transcriptional upregulation of PDL1 and PDL2 during the immune response, in response to interferon gamma. So basically, at the site of inflammation, interferon gamma that is produced by T cells induces activation of JAK1, JAK2 kinases, which phosphorylates which phosphorylate JAKs, STATs, and STATs go to the promoters of their responsive genes and upregulate their expression. One of such genes are PDL1 and PDL2. The amplification of this locus was also associated with activation of JAK2, at least in those cell lines which had the highest copy number gains. As you can see here on the right in these four lines on the right. So the question we asked was whether by inhibiting JAK2 can we decrease expression of PDL1 and PDL2. So to answer that question, we used an inhibitor, JAK2 inhibitor that was at that time in clinical testing. The inhibitor acts on target by decreasing JAK2 phosphorylation, and in parallel that was associated with decreased transcript abundance of PDL1 and PDL2 as well. So the single amplification that we identified in Hodgkin lymphoma provides basically two disease-specific mechanisms that work in cooperation to facilitate immune escape of the tumor. First lesion is amplification of PDL1 that is directly responsible for immune escape, and this is a targetable lesion with the antibodies as we know now. But the second lesion that is created by this amplification is actually activation of JAK2, which further fuels overexpression of PD1 ligands, but in parallel it is responsible for increased proliferation. It is also targetable lesion, so we can target the pathogenetic mechanism associated with this amplification with specific drugs. As you know, a couple of years later, these studies led to a clinical trial with PD1 blocking antibody nivolumab that basically demonstrated that it is a highly active therapeutic strategy in these patients. Hodgkin lymphoma patients, after multiple relapses, after allogeneic transplants, still respond to nivolumab, and this is a rescue treatment these days for these patients. Okay, so if we think more about this immunotherapeutic or mechanism basically responsible for immune evasion, we already identified two, but there is much more. If you think about this, there are cytokines that are produced by tumor cells that are responsible for creating this immunosuppressive microenvironment. There are cytokines that are responsible for Th2 polarization, and there are molecules that are induced in tumor cells that are directly immunosuppressive, such as GAL1, GAL2, and PDL1 and PDL2. So even by targeting one of these mechanisms, we still leave all the others intact and operative, right? So we wanted to identify a mechanism that would be targeting at least not one, not two, but more of them, right? So we thought, what could these protons listed here have in common? And when we thought about that, it occurred to us that they are actually all regulated by constitutive activity of two transcription factors, NF-kappa B and STATs. Unfortunately, these transcription factors are very poor therapeutic targets, as you know. In general, transcription factors are not good therapeutic targets. So, another question. What is the upstream regulatory mechanism that could control both of these transcription factors that would be targetable? And then we identified PIM kinases, PIM1, PIM2, PIM3 oncogenic kinases that are actually oncogenes responsible for many tumor-specific functions such as metabolic functions, protein translation, cell cycle, control of apoptosis, but they also regulate oncogenic transcription factors, including STAT3 and NF-kappa B, but also c-Myc and FOXO. And as kinases, they are good therapeutic targets. So we developed an inhibitor of these kinases in collaboration with Cellvita, a biotech company from Krakow, to study the function of PIM kinases. But before that, we first looked whether PIM kinases are in fact overexpressed in Hodgkin lymphoma cells. So we stained a series of primary tumor sections and identified either overexpression of PIM1 or PIM2 or PIM3 or the combination of the three in 97% of these patients. So basically, it's universally expressed in this tumor. And this beautiful picture appeared on the cover of Blood last year. So that's the credit of our hematopathologist who performed stainings. So can we target PIMS and can we trigger expected immunoregulatory results with inhibition of the kinases? So first we evaluated the activity of the compound, and first we noted that the pan-PIM inhibitor inhibits activation of STAT5, inhibits activation of STAT3, decreases phosphorylation of REL-A P65, which is a subunit of NF-kappa B, and also decreases protein translation. So we have on-target activity, and we have the decreased activity of the transcription factors that we know control expression of multiple immunoregulatory proteins. So we silenced, to control the activity of the compound with genetic methods, we also silenced PIM kinases with siRNA, and we assessed the expression of galectin-1 and PDL1, PDL2 in this tumor. And that, as you can see here, by assessed by Western blot, we have decreased expression of galectin-1, and we have decreased expression of PDL1 and to a lesser extent PDL2. Then we repeated this experiment with the inhibitor that we developed. And here are the results. We have decreased expression of galectin-1 and massively decreased expression of PDL1 and PDL2 in this tumor with this inhibitor. In addition, we have decreased expression of NF-kappa B controlled cytokines such as TNF, IL-8, CCL5, IL-13, and CD40. So another evidence that the activity of this compound leads to potential immunoregulatory effects. Finally, we compared, we assessed the activation of T cells that are co-incubated with Reed-Sternberg cells that were exposed to PIM inhibitor. And we noted that the cells that are mixed and co-cultured with PIM inhibitor T cells exhibit higher activation markers, so CD69 and CD25 expression. So they basically are more activated when the cells were pre-exposed to PIM inhibitor. Finally, we looked at the activity of the compound in the in vivo model. We were aware of all imperfections of this model. This was a xenograft model, right? So there is no immune system in this tumor. We know that, but there is no better model. As I said, we didn't have a syngeneic model. We failed creating it, but still we could derive some conclusions from that model. So first we observed that the compound basically delayed or arrested the tumor growth in these mice. So the bottom line shows the growth kinetics in mice treated by the compound. The upper curve shows the growth kinetics in control mice. And then we explanted tumors and assessed the expression of these immunoregulatory molecules that I mentioned. So galectin-1, which was basically wiped out from the tumor, and PDL1, which is also decreased. Phospho-S6 is basically is a target engagement biomarker. Phospho-S6 is very specific to demonstrate that PIM actually do their job at the tumor site. So it also the inhibitor also erased this phosphorylation in tumors explanted from the mice. So to summarize this talk about immune evasion mechanism in Hodgkin lymphoma that we identified, we first show that classical Hodgkin lymphoma Reed-Sternberg cells are hardwired for immune evasion, either by functional mechanisms that lead to overexpressions of certain molecules, or by structural alteration directly leading to overexpression of PDL1 and PDL2. We showed that galectin-1 is an immunoregulatory protein that is expressed by classical Hodgkin lymphoma Reed-Sternberg cells. They are expressed, we showed that PDL1 and PDL2 checkpoints are overexpressed in Hodgkin lymphoma due to copy number gains So this is a 9p24 locus, which provide a clinically available target, or actually two targets, because JAK2 is another complementary target in this aspect. But we also identify very complex and redundant mechanisms responsible for this immune evasion, and this highlights the need to develop pleiotropic therapeutic strategies that would target them simultaneously. And we identify PIM kinases as a potential therapeutic target that meets this requirement. So it controls the activity of transcription factors responsible for induction of immunoregulatory proteins, cytokines, chemokines, galectin-1, and PD-L1 and 2. And we also demonstrated therapeutic immunomodulatory potential of PIM inhibitors in classical Hodgkin lymphoma. And finally, I would like to thank all the collaborators that were involved in these studies. First of all, Michael Grimm from MD Anderson Cancer Center, and previously from University of Nebraska Medical Center. My colleagues from Dana-Farber Cancer Institute, colleagues from Medical University of Gdańsk, Maciek Zaucha, my collaborators from my institution, Institute of Hematology and Transfusion Medicine, from the Department of Diagnostic Hematology and Department of Hematology and Department of Experimental Hematology, colleagues for Celvita who developed the inhibitor, and Center for Postgraduate Medical Education who helped us with the xenograft models. Last but not least, our funding sources was Leukemia Lymphoma Society of America and Polish National Science Center. Thank you very much, and I'll be happy to take questions.
So we'll start discussion, and maybe I start with one comment and one question. So my comment deals with nivolumab. We know how potent this substance could be, but you said that it is active in terms of clinical setting of auto and allogeneic stem cell transplant. In terms of allogeneic stem cell transplant, we have just recent report that nivolumab could induce GVHD, so I don't think that is clear setting. So
yes,
after autologous transplantation is clear,
right.
But after allogeneic, I think the modulation of immune system is not so easy, and this intervention might be
excellent point.
Requires more
absolutely
details and
but it doesn't mean that it's not active against tumor, but
no.
it also has basically has undesired side effects, right?
Yeah. And the question concerns PIM kinases and PIM inhibitors. In your earliest studies, you compared Hodgkin lymphoma with DLBCL, which is very nice system because both are aggressive lymphomas with different influence of immune system. So this system is perfect, seems to be perfect. But PIM inhibitors are also overexpressed in the DLBCL, and you know that some studies show that PIM inhibitors in terms of DLBCL were not active because there were several intrinsic resistance mechanisms induced during therapy, and also in AML the same story. So the ex vivo models doesn't work, and in fact the first clinical trial was stopped. So what's your comment? Why you think that still PIM might be good target in terms of Hodgkin lymphoma?
I think it can be a good target in general, but you have to think carefully who should receive it and what are the good biomarkers for that. In terms of that earlier studies on PIM inhibitors, I'm not really convinced that the inhibitors used were the right ones. None of them made it to clinical trials, or even those who did it the trials were terminated because of target toxicities. That was with AstraZeneca compound AZD1208, which was in clinical trial in AML, but due to cardiotoxicity, which was related to an off-target effects to some potassium channel, was the off-target so that was the mechanism of this cardiotoxicity. The trial was terminated prematurely. I think that the studies in Hodgkin, in the diffuse large B-cell lymphomas that are available, basically there are two major conclusions from those studies. First is that PIM PIM kinases are markers of the progression in this disease. So the more aggressive disease, the higher expression of PIMs in this tumor. And in this paper, that was an oncotarget paper, like published 4 years ago, maybe. I don't remember exactly the inhibitor they used, but it was some sort of a tool compound that didn't show really much activity in this tumor. We, of course, diffuse large B-cell lymphoma, for those of you who don't know, is the main focus of my lab. Hodgkin lymphoma is some sort of a side project, but it turned out to be a pretty nice story that was published well and had good consequences for the patients. But we also, of course, started with diffuse large B-cell lymphoma, our experiments. It's a potent compound, active in vivo model as well. And we have very specific mechanism that could be responsible for the activity of this drug. It's not related basically to all known targets of these kinases, so not translation, not transcription factors, but basically epigenetics. I don't want to say too much because it's a very early project. We are in the process of evaluation of several grants that would facilitate us continue those studies, but I think that it can really lead to a very nice mechanism, really nice stories about PIM kinases in diffuse large B-cell lymphoma. So I believe it is a good target. The question is whether who should receive it. And the complication in diffuse large B-cell lymphoma is stems from the fact that PIM kinases are very common mutation targets in this disease. So about 30% of patients have mutations within PIM kinase. What is the reason? We know the reason. The reason is AID activity, activation induced deaminase, which is one mutational mechanism in this disease. We don't know what it does, whether it makes the tumor resistant to inhibitor, whether it makes the protein more active. We don't know that. We want to examine that and study that in more detail to know that. But most importantly, about 30% of mutations of PIM kinases in DLBCL are not derived from AID activity, but from aging. So these are spontaneous deamination. So this is strictly oncogenic age-related process. I believe that it's meaningful. I don't think it's by accident. P53 is mutated from aging. Many RAS is mutated from aging. So I think that this is a potent oncogene that can be studied that way and can provide very important insights to the mechanisms related to this disease. With respect to AML, can I still take some time or not?
Just sum up this in 1 minute.
Dominika has a question.
Please sum up only the AML.
AML, we have this compound. This compound is in clinical trial.
But trial was stopped.
It was halted due to drug-unrelated death of the patient, but it's continued now.
It's unrelated to drug.
Yeah, so the patient was receiving like 23 other.
We know how AML is difficult as a disease to be targeted.
Yeah, so the trial is continued, and I cannot disclose any results of this study yet.
To have question.
Mm-hmm. 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 It's a good question because the simple answer is that would be my first guess, but after a deeper thought, I'm not really convinced, because if you decrease PD-1 expression, then you may not be, you may basically create antagonism, actually, not a synergy, because you decrease the target. On the other hand, by decreasing the expression of PD-1, you may need less antibody to block it. So both possibilities are actually testable questions, yeah.
But would you combine this with downstream molecules like PD-1 or Galectin-1, or better different pathway like JAK1, JAK2?
I don't think that blocking different pathway is a good idea, especially JAK1, JAK2, because actually we block JAK2 with this inhibitor. So I would rather guess that the PD1 or PD1 blocking antibodies like nivolumab would be better, and they are available clinically, right? So that would be the first thing to test, because that the tools are available.
Okay, thank you. Tomasz Cogosa.
So so this, to make a long story short, the galectin-1 antibody patent was sold to Medarex, and nothing happened with it after that. So I don't know what's the policy of the company. We didn't get any feedback. I know that galectin-1 guru Gabriel is still developing small molecule inhibitors of galectin-1, and yes, it's still alive. Yeah.
Professor Młynarski, last question.
Mm-hmm. Because you're looking where the light is. We are looking at the expression of galectin-1 in the tumors. PD1. PD1, main source of PD1 are macrophages in this disease, and it was published in JCO last year. And the yeah. Yeah. Mm-hmm. I am not an expert on lung tumor, so I'm I'm not going to answer this question. But importantly, we have a grant in the lab. Maciek Szydłowski, who was the first author of this Hodgkin lymphoma PIM study, noted that PIM kinases are also expressed in macrophages by immunohistochemistry. Then we'll try to differentiate human monocytes to M1 and M2 macrophages, and PIMs are expressed after differentiation into M2, not in M1. So they are present in this bad immunosuppressive macrophages. So by targeting PIMs, we probably would also inhibit them in macrophages. But what are the consequences of inhibitions of PIM in macrophages? We don't know, and this is the goal of this grant.
Okay, thank you. The next speaker is Professor Wojciech Młynarski. We now move to pediatric ALL field, and we will see what are possible molecular targets.
So actually, thank you very much for have this opportunity to share with you some of our data. This presentation will be more clinical than basic science, but definitely I can show you some data which were generated in the international collaboration. Why? We need to have international collaboration with very specific disease like acute lymphoblastic leukemia, because this disease is very rare as compared to Hodgkin lymphoma and other diseases. This is unusual disease. So basically, this disease is very unusual. But if you look at the cancer type among the pediatric patients, you see here that leukemias are the most frequent diseases, and among acute leukemias, acute lymphoblastic leukemia is the most important and the most common disease. So, for instance, in Poland, if you consider 2016, if you have almost 200,000 patients diagnosed with cancer, only 1,000 was diagnosed among pediatric patients, and about 200 individuals, pediatric individuals, children, suffer from acute lymphoblastic leukemia. So, in general, this is really rare condition, and this is a very heterogeneous disease as well. So this is why we need to put any very solid statement or solid evidence that some treatment or targets can be efficient, we need to have very large numbers in the field of international collaboration. But first of all, we need to have, based on the collaboration, some genetic background of the disease, because as I said, this is very heterogeneous. So we know right now that among pediatric patients, you have really progress in the treatment options. So, for instance, these are the data from our department in Lodz, and as you see, in each 5-year interval, there's an increasing improvement of the treatment patients, all patients with cancer. If we look specifically at the acute lymphoblastic leukemia, right now most of the trials are finishing at the level of about 90 to 95% patients who are cured from this disease. So it means that acute lymphoblastic leukemia is almost curable disease among pediatric patients. But of course, there is about 10 to 15% of patients with acute lymphoblastic leukemia who suffered at least relapse, or some of the patients are resistant to our treatment. And what kind of risk factor for ALL relapse we have? We have some clinical evaluation, like initial poor steroid response, because in most cases we use the first drug as for the treatment of acute lymphoblastic leukemia is steroids, and we start with the steroids for 1 week, and after 1 week we can establish the response to treatment. And if you have not efficient response to this just one single drug, it means that probably we'll have the problem with the patients later on. Of course, white blood cells, it's another marker which also corresponds only with the number of cells in the body. Phenotypic features, immunophenotypic features like origin of the cells, B or T. Of course, age is a very important factor, not only in pediatric population but also in adults. MRD, it means minimal residual disease, which can be established using flow cytometry or molecular methods, even NGS, but the last one is not really standardized method yet. And of course, what I try to target during the lecture is the genetic abnormalities found in the leukemic cells. So before era of the genome-wide studies, in general, using even in current protocol for treatment of patients with acute lymphoblastic leukemia in Poland, we have just 3 genetic lesions which can be considered as a risk for the patients. So we have large spectrum of different genetic defects observed in acute lymphoblastic leukemia. This is still the picture from the before genome-wide studies. And based on this, we can consider just hypodiploidy. So too small number of chromosomes, less than 42, which are very unfavorable prognostic marker. BCR-ABL, this can be observed mostly among this fusion is mostly observed among patients with chronic myelogenous leukemia, but also in some percentage, a very small, tiny percentage, as you see, that just 3% of the patients with acute lymphoblastic leukemia can have this lesion, and this is also poor prognostic markers as well. And rearrangement of the another gene, MLL, which can occur among the patients, among the neonates, very early developed leukemia, mostly even leukemia generated during the first month of age. And also this can occur this lesion can occur also among the patients with secondary leukemias, and we treat these leukemias very seriously because the secondary leukemia are really poor response to our treatment. But based on these lesions, we can target only about 15% of the patients who has some kind of unfavorable outcome. But we know that there are many other patients who developed relapse or are resistant to treatment, primarily resistant to treatment, and we are not able to figure out why they are not respond to our classical treatment methods. And I can show you one case. I think it's a very elegant paper presented by Gianni Casaniga some years ago during the ASH meeting in New Orleans. He just showed the twins, monozygotic twins. Both of the twins developed leukemia. Both are positive for BCR-ABL fusion. And as you see, both these guys developed leukemia in very similar time point of age. But unfortunately for the one of this guy, first has a very good response to treatment, even to steroid, the first type, very low MRD level, and he's still in the remission. I have a contact with Gianni Casaniga because we are in the collaboration right now to look for other lesions can be targetable for in pediatric ALL. But another child, completely the same, looking at the constitutive genomics, has poor steroid response, has high level MRD, and resistant to treatment, and this guy died, unfortunately. And the only one difference between these two was genetics of the leukemic cells. Hyperdiploidy in the one case, and the Ikaros deletion in another one. And this is a very nice example that even the same genetic background can differ at the development of leukemia. So, for instance, you have both these children, both these twins, boys, they have the same lesion at the very beginning. So the primary hit was completely the same, but the outcome and the outcome was the same because they both developed acute lymphoblastic leukemia. But unfortunately, one of them has kind of very unusual and deletion which is very unusual for the patients at that time, and this guy not respond to our treatment. So starting the history of the Ikaros from this point, there are plenty of papers showing that Ikaros lesion in acute lymphoblastic leukemia is very important part of the genetics or genomics of acute lymphoblastic leukemia. What we have in what kind of lesion we have in the gene of Ikaros. So basically, in most cases, we have a deletion, intragenic deletion, which occur between exon 4 to 6 to 7. And this gene is lacking of the DNA binding domain. Unfortunately, this can generate heterodimer with the normal part of the normal allele of the gene, and finally develop a kind of dominant negative effect for the leukemic cells. If you look at the expression, you can see here that wild type have, because this Ikaros is a transcription factor associated with the differentiation of lymphocytes, especially in B cell lymphocytes. So if you have wild type, you have very nice expression in the nuclei. If you have deletion, you have just expression in the periphery, not in nuclei. And if you have, of course, a biallelic null mutation, you have no expression at all. In the monoallelic, you have reduced expression in the nuclei, so it means that you have a reduction of the function of Icaros. And because the most frequent lesion in acute lymphoblastic leukemia is the deletion, it means that basically the patients who has a deletion has a dominant negative effect and no Icaros function. And from the perspective of the cell, it stops the differentiation of the cell and switch the cells into very immature phenotypic features which are unresponsive to some of the drugs, especially asparaginase, which is one of the important drugs for the acute lymphoblastic leukemia treatment. So after these discoveries, many papers occurred that Icaros is important from clinical point of view, and about 25% of patients with AL has deletion of Icaros. So it, as compared to pre-genomic era, pre-whole genomic era, we have just, before the era, we have just 15% lesions which can be diagnosed as poor responding. Based on just Icaros, we can add 25% of the lesions which probably can serve as a surrogate of poor response to treatment. If we consider this in pediatric population only, you can see that patients who has the deletion of Icaros have very high MRD and also has very high relapse rate as compared to patients who has no this lesion. We still, as you know, we are right now, we are playing with the patients with just 10% of the patients who are not be treated or cured from leukemia in general population. The same if we consider just very high risk group of patients based on the stratification models. So basically, if you have deletion of Icaros, it's something very often something wrong happen with the patients. The same in the standard risk groups. These are another paper published in the collaboration also with our group. That if you have patients with a standard risk who in general are treated not very seriously and risk of relapse in this group is really very low, so it means about 3 to 5%. Even in this group, you can, if you find the patients with Icaros deletion or a point mutation, you can extract the patients who probably developed relapse later on. Interestingly, if you look at this very tiny subset of acute lymphoblastic leukemia called BCR-ABL positive. As you remember from my talk, it is just 3% of the patients. So it's really a low percentage of the patients. Among even this group, if you analyze Icaros deletion or Icaros lesion in this group, you can find Icaros in about almost 90%, and these patients are at risk of relapse. So within the group who is very poor from a prognostic point of view, you can consider even poorer group based on the just one lesion, extra lesion like Icaros. And as you see here, that these patients are really poor from prognosis, as you see here, for instance. But the question was whether the Icaros mutation is a risk for relapse in all patients with acute lymphoblastic leukemia, especially pediatric patients. And this is not really straightforward because if you look at the different abnormalities found right now in acute lymphoblastic leukemia, you can have one player, ERG, mutants, ERG deletion. This is also, as you see, very a tiny percent of the leukemic patients. But among these patients, if you have Icaros deletion, there is no, and ERG deletion, you have no effect of Icaros. So this is, from clinical point of view, it's really important, because if you would like to, if you have most of the lesions which can observed in the acute lymphoblastic leukemia, and try to establish new protocol for treatment of patients, for just new lesion, which can be included in the protocol, can be always confirmed by many centers, many study groups, that this is the risk factor to be considered in the future protocols. So it's not a kind of exploratory, but you need to have confirmatory approaches to put the new lesion as a marker for the treatment, and to target the treatment later on. So right now we put this Icaros group. This is very recent paper published in JCO by Martin Standula, which was confirmed also in different groups, that if we try to consider the risk group based on the genetic defects and Icaros, we need to consider Icaros plus other defects, as you see here, and no ERG deletion at all. This is the incoming protocol. So probably we'll start the treatment also Polish patients with this using this protocol within the few months from now. We are trying to begin at the early October. And in this protocol, there are very difficult stratification options, including some genetic defects and other. Basically, the most important issue is that we will include this Icaros Plus group in the stratification, and to find in this group another option of treatment. This was also confirmed for a Polish cohort, that Icaros deletion together with CDKN2A deletion is a risk factor for relapse of ALL. But basically, this is the story of Icaros. It's not finished yet. If we consider Icaros deletion, probably this is more complex, and if we were focused on the other lesions found in the lymphoblasts, in leukemic cells, but in other groups in Memphis and also in Rotterdam, they studied expression profiling of the cells, of leukemic cells, to develop, to find another subtypes of leukemia. And based on the expression profiling, they found another subtype of leukemia with the expression pattern very similar to the pattern observed in this unusual leukemia BCR-ABL. So they call it BCR-ABL-like because of the looking at the expression level. And as you see, this group of patients, if you consider outcome, they have really very poor outcome, similarly to BCR-ABL leukemia. And what we have as lesions in this group, we have also Icaros defect. So it's even more complex. We have Icaros is a kind of another marker which probably is somehow linked with the another subtype of a disease. But this subtype of the disease we are not able to extract based on the genetic lesions, but only based on the expression profile. So from clinical point of view, it's really difficult because you should analyze all the patients. Actually, you need to have, for each patient, you need to have expression profile for this one case and to compare to another one. And this Phi-like or BCR-ABL-like group has really a poor outcome. And unfortunately, this is something interesting, especially for adults and hematologists, and also for pediatric hematologists, because the frequency of this disease is very low among pediatric small children. This increased during the time, and later on, there's a peak for young adults of the frequency of this BCR-ABL-like, and later on, you will have only or mostly patients with chromosome Philadelphia leukemia. So it means that teenagers and young adults is a group with a very high frequency of BCR-ABL-like phenotypic features. So it means that this group is a very probably this is why this group of patients has a poor outcome. And as you see here, among pediatric patients, this is just about 15 patients among the, for instance, young adults. You have about 30% of the patients with BCR-ABL. But if we look at the BCR-ABL-like group, we found that most of the patients has kind of different lesions which activate different pathways related to the kinases. This is somehow good news for the patient's point of view, because most of the lesions can be targetable. And if you look at the targetable therapy among patients with acute lymphoblastic leukemia and BCR-ABL-like phenotype, it seems that we have at least 2 drugs on market which can be used for the treatment, for extra treatment of patients with these phenotypic features. And right now we know that this can be really efficient in some cases. So this what I show you, this was collaborative work, ours together with our centers. But right now I will focus just on a few cases. Of course, you need to consider that this case is the patients, just human being, and all of these patients without a targeted therapy will not survive. So based on our experience from our lab, we generated a few years ago the Central Laboratory for Genetic Testing for Pediatric Cancer as a national service, especially for acute lymphoblastic leukemia. And I share with you some of the cases which we found based on the analysis of the patient's genome. So for case 1 is I am personally involved in this case because during my overnight duty, this patient was admitted with the relapse, and as you see, very high number of white blood cells. His primary diagnosis was in 2013, so 3 years before, currently relapsed. And we found in this patient the deletion of Icaros and CDKN, so it's kind of Icaros plus group. But unfortunately to him, we found also fusion with another gene. And we can say that this patient suffered from BCR-ABL-like disease. Unfortunately, after first block of the chemotherapy, or even 2 blocks of regular chemotherapy for relapse, he has still very high number of cells, so it's not responding to the treatment, which was really heavy treatment. And we found First of all, we found one case in the literature similar to our, and we also found that this fusion, that this is unusual fusion, and this is extra chromosomal fusion. It's not located in the chromosome, but in these cells, leukemic cells in the patients, you can observe these lesions outside. You have this circular DNA fragment like plasmids in the human cells, which the number of these plasmids depends on the severity of the disease, or also activation of the kinase pathway, ABL1 kinase pathway. And based on the experience of the Italian group, we treat the patients together with the chemotherapy with imatinib. It has to target this molecular lesion, and right now the patient is still alive after transplantation and in good shape. I share with you another case published in Hematologica during our case was treated. Completely the same story. So I'm sorry, there are some Polish words over here, but basically this patient was relapsed, was unresponding to the treatment, and was transferred to palliative care. Fortunately for him, they have the material from the blast, and they reanalyze genetic lesions in this patients. He was again admitted to the hospital, and the treatment with TKIs, tyrosine kinase inhibitors, and he's alive. Another case from Gdańsk, no remission, so this is really severe. If you have the pediatric patients with ALL with no remission at all, after 33 days, it means that the risk of unfavorable outcome, death, is about 99%. This guy, even at the day 52, has still no remission, so it means that the leukemic cells were resistant to our treatment. But fortunately to him, we found that this guy has a higher expression. This is unusual also, expression of the CLF2 on the surface of the leukemic cells. We found in our very recent paper that the patients who has this overexpression of the CLF2 on the surface of leukemic cells, they have very often activation mutation in JAK2. And in this case, we found the mutation, the fusion of JAK2, which activated the JAK2 kinase. And we gave this guy ruxolitinib for the treatment. Unfortunately to him, he has even sepsis during this treatment. We stopped the treatment, and later when we get back to the treatment with just 2 drugs which were already done in the previous treatment, and just these 2 drugs generate the remission of the patient, and right now the patient is also in good shape after the treatment. As you see, addition of the ruxolitinib, molecular treatment, generated the remission of the patients. And this corresponds to what we have right now as a collaborative group at European level. We have this new protocol. Many of the lesions can be targetable for the patients who are unfavorable outcome. So basically, we need to have this kind of lab in each country to look for the lesions, molecular lesions, which can be targetable by the different drugs. So, for instance, FISH and also all this analysis will be done for each patient who are not responding well for standard therapy. So it means that if right now we have about 90% chance to cure the patients with active lymphoblastic leukemia, if we add all this stuff on board, probably we can go even further with the improvement of the treatment. And this is the acknowledged for all the collaborations from Poland, and then also from collaboration from our institution also institution abroad. Thank you very much for your attention.
Thank you. For the sake of time. Because you said deletions, but what's the nature of those deletions? Are they biallelic or monoallelic?
So I showed this in the presentation. So mostly this is monoallelic.
And the second allele is mutated, possibly? No.
It's intact, but this is the dominant negative effect, because you can have generated two different proteins, one without transactivating domain. So if we put this as a heterodimer, this protein will stay in the cytoplasm, not.
Okay, so
not
it's haploinsufficiency.
So it is a haploinsufficiency, but with dominant negative
okay.
effect.
Thank you. And now we turn to oral presentation, the best abstracts that were chosen for the presentation. First is by Artur Wnorowski, development of MNF as inhibitor of pancreatic tumor growth. So we have 10 minutes. No. So your slides should be downloaded on the. So maybe do we have?
What I would like to show to you is our profiling of metastatic cells and different stages of metastatic cascade in breast cancer and their clinical significance. So you probably know that breast cancer is one of the most frequent cancer in women worldwide. And what clinicians fear the most is the development of distant metastases, because once they develop, the prognosis is poor. But we also know that before metastasis develop, the distant metastasis develop, tumor cells can also be found in regional tumor draining lymph nodes, and also in the circulation as the circulating tumor cells. So the circulating tumor cells, which we are working with, are the cells or very rare cells in the blood of cancer patients, maybe 1 to 10 per 7.5 milliliter of blood. They were first described by Ashworth in 1969, and for over 100 years, nobody was paying much attention to them until it was the test for searching for detecting circulating tumor cells was approved by FDA. It was Cell Search, and it was shown that more than 5 CTCs per 7.5 milliliter of blood is related to poor prognosis of the patient. Later on, similar things were shown also in early breast cancer patient, and the interest in CTCs spiked significantly. So analysis of CTCs is fairly easy. You can just take a blood sample from a patient, fairly non-invasive method, and then enrich the sample for your CTCs of interest. But once we started working with CTCs and we gained more information about tumor biology, we found that the process of epithelial to mesenchymal transition is involved in progression of cancer. So what the EMT is? It's a physiological and physiopathological process that changes morphology of tumor cells, or not only tumor cells, but epithelial cells, which are fairly immobile, restricted, polarized, to more motile cells, like mesenchymal cells. And those cells are able to degrade extracellular matrix. They can travel, maybe they can travel to the nearest blood vessel, lymphatic vessels, and they can form metastasis more efficiently. What's also important that this EMT transition changes significantly the profile or molecular profile of tumor cells. So epithelial cells, which normally express epithelial markers such as cytokeratin, 19, E-cadherin, they lose this marker and they acquire mesenchymal markers like vimentin on N-cadherin. And what is important here for CTC's isolation, that cells after EMT cannot be detected because the currently used method, these FDA-approved methods, are targeting epithelial markers on the surface of CTCs. So if they are lost, we cannot find them. So when we wanted to study CTCs, we had to come up with a different approach. So normally we would catch only those epithelial cells red, but we would totally miss the mesenchymal cells, which might be the really malignant cells. So we came up with a different approach. We used immunomagnetic negative selection where we removed blood cells from the blood sample to enrich all the CTCs present in the blood sample. So the general aim of our study was to characterize EMT profile of tumor cells at different stages of the metastatic cascade. We looked at the primary tumors, at the CTCs and lymph node metastasis, regional lymph node metastasis, and checked additional genes related to invasion, metastasis, and cancer stem cell phenotype. So we analyzed, collected a group of 107 breast cancer patients, early breast cancer patients. We had primary tumors, circulating tumor cells, and 50% of those patients had lymph node metastasis. Additionally, we collected blood samples from healthy controls to correct for the level, basal level of these EMT cancer stem cells, CTCs, in healthy controls. So our CTC isolation method was fairly simple. We took a blood sample, did gradient centrifugation, enriched sample was then incubated with CD45 cover magnetic particle to deplete the sample from blood cells, and then such sample was analyzed for gene expression. We tested genes related to presence of CTCs, EMT markers, EMT transcription factors, invasion metastasis related genes, and also cancer stem cell markers. So when we looked at the enriched CTCs population, we did show on immunofluorescence, maybe this is not a good image, that we do see them. We find cytokeratin, mammaglobin positive, vimentin positive cells, and we defined by qPCR presence of CTCs as expression of one of these mammary epithelial transcript, mammaglobin, HER2, plus cytokeratin, and this was called an epithelial CTCs, or presence of epithelial CTCs, or if vimentin was present above the threshold level, we said we have mesenchymal CTCs. So overall, we found epithelial CTCs in 17% of the patient, mesenchymal in 13% of the patient. So from the early breast cancer patient group, we find CTCs in at around 30% of the patients. And then when we looked at the gene expression analysis, we found that those mesenchymal markers were actually increased together with markers related to invasion and metastasis. CXCR4, which is a receptor thought to be involved in metastasis or attracting tumor cell into the metastatic side, or UPAR, which is a receptor involved in degradation of the extracellular matrix, was increased in mesenchymal CTCs in comparison to epithelial CTCs. When we looked at the expression of stem cell markers, we also noted that almost all of the markers were increased in mesenchymal CTCs. What is also important, the prognostic role of CTCs. And we show that not only the presence of CTCs, but their molecular or their phenotype is the most important. So here you can see that these are the mesenchymal CTCs are the bad ones, in comparison to epithelial CTCs, which were linked to actually better prognosis than mesenchymal ones. In the multivariate analysis, we show that presence of mesenchymal CTCs is linked to almost 6 times higher risk of death, and was not related to T stage or N stage of the patients. Then we had a look at the primary tumor and the lymph node metastasis. We showed that signs of EMT activation in primary tumors in 43% of the cases, in lymph nodes in 25% of the cases. And when we look at the prognostic significance, we actually did not see any correlation between expression of EMT markers, activation of EMT, and the primary tumor. But in the lymph node metastasis, high expression of EMT markers, transcription factor, TWIST1, and SNAIL was related to poor prognosis. Then we assessed custom stem cell markers in primary tumor lymph node metastasis and also their heterogeneity. So we took 5 spatially separated areas of the primary tumor and lymph node metastasis and analyzed them on tissue microarrays by immunohistochemistry. And what we found that actually in comparison, or primary tumor and lymph node metastasis had fairly similar, apart from ALDH marker, there was similar level of the stem cell expression. But when we look at the heterogeneity, measured as a heterogeneity index, in that case it was a Gini index, which is used in economics, for example, to calculate inequality in income, on the scale of 0 to 1, where 0 is the lowest heterogeneity, 1 is the maximal heterogeneity, we found that in the lymph node metastasis, heterogeneity is much more decreased. So we actually think we observe a selection of these malignant clones from the primary tumor to lymph node metastasis. When we also now divided sample into epithelial and mesenchymal phenotypes and also looked at the stem cell marker expression, here we didn't see such striking differences as we observed in CTCs. So only in case of OCT4, the stem cell marker, we noted that elevated expression of this marker is occurring in mesenchymal samples. So to summarize, we think what we are observing is the selection of the malignant clones during metastatic cascade. So cells from the primary tumor have to go through this bottleneck, and the ones that are able to escape, so CTCs or lymph node metastasis, are enriched in these malignant features, especially the mesenchymal phenotype we think is crucial here. It's involved in more aggressive phenotype with stem cell phenotype and also poor prognosis of the patient. So profiling these populations gives us more information than profiling of the primary tumor, which is very heterogeneous, and where finding those aggressive populations might be quite difficult. And considering that actually in CTCs we found the strongest significance, we wanted to go a step further. So we know that actually CTCs are rare. We know that they are heterogeneous, so actually the only way to know their biology in detail is actually to go into single cell level. So we developed a method to profile individual CTCs and look at their biology. In the test experiment that we are developing, or that we developed, we spiked epithelial or mesenchymal breast cancer cell lines into blood sample, processed the samples, and then picked individual positive cells into a test tube, and then mRNA isolation, preamplification, and gene expression analysis. And we showed on these epithelial and mesenchymal markers that we can easily discriminate the phenotypes. So we can capture just a few circulating tumor cells in the blood sample. We can tell what's their phenotype. And now the next step will be to add additional genes and look at the biology of CTCs from breast cancer patients. So here I would like to thank you and acknowledge also the patients who kindly donated their material for the studies and everybody involved in the research. Thank you.
Congratulations. And questions?
I think it's all parallel. And there's a recent paper, actually from this year, from a month ago, that shows in mouse model that lymph nodes actually also metastasize as well. And lung metastases that were found in mouse model, breast cancer mouse model, were actually predominantly coming from the lymph nodes. So the lymph nodes can also seed into the circulation. So we really don't know. I mean, there's not enough data about that, and I think our research is also aiming to answer that question. I mean, if we had access to frozen lymph nodes, we could also analyze living single cells, in the lymph node, compare them to the CTCs, to the primary tumor, and then maybe we could see the bigger picture. But there are still very technical limitations, and I think nobody have done that before.
Do you plan to implement cell-free DNA analysis into your system?
Yeah, we are thinking about that, but the blood samples that we are collecting at the moment, we are planning to also to use for cell-free DNA analysis. So we are going to, take a serum sample and bank it, keep it maybe for later, and see what comes out from the CTC's analysis, and then look in parallel later for something in cell-free DNA.
Okay, thank you. The last speaker is Dr. Norowski, now with presentation.
Okay, so I would like to thank for this opportunity to present my research, and actually I would like to tell you a short story on early stage of cancer, of drug development project, and the main character of the story is our leading compound, formitox-1 naphthalophenolterol, or MNF. And in this project we focused on pancreatic cancer, which is in the US, it's the 4th leading cause of cancer-related death, and it's predicted to become 2nd by 2030. And pancreatic cancer has one of the worst prognosis, as only 6% of patients will survive beyond 5 years post-diagnosis. And this poor survival indicates the need for development of new drugs, including target-specific agents. And we are lucky to have RRMNF as a selective agonist of beta-2 adrenergic receptor. So this compound was developed as one of nearly 100 derivatives with an aim to get a compound that will specifically target beta-2 over beta-1 adrenergic receptor. And as a proper agonist, it activates the receptor to trigger signaling pathways dependent of this receptor, including GS, GI, and beta-arrestin. Later on, we discovered that apart from activating beta-2AR, which was quite beneficial in our in vitro assays in inhibiting growth of cancer cells, that RRMNF is also an antagonist of another receptor, of GPR55. And this fellow is an recently deorphanized G-protein coupled receptor that is sensitive to cannabinoids and lipids. However, its physiological function is actually undetermined yet. But we know that when this receptor is overactivated or overexpressed, it drives the proliferation of cancer cells, makes them more aggressive. So with RRMNF, we can block downstream signaling downstream of this receptor. And we carried out some initial in vitro experiments in human pancreatic PANC1 cells, and we observed dose-dependent inhibition in proliferation with sub-micromolar efficacy. We also looked at some markers of proliferation. So we observed decrease in phosphorylation of AKT kinase in response to RRMNF and to another antagonist of GPR55. We also looked at some classical cancer biomarkers that were also suppressed upon treatment with our compound. So we wanted to move to in vivo experiments to confirm these in vitro observations. So we started a xenograft study in mice with PANC1 cells that we implanted into the animals and subjected them to either vehicle control or RRMNF. And we observed the growth of the tumors, but actually there was no effect of this compound, so we were unable to recreate the in vitro effects in an in vivo model. So we began to wonder what went wrong. And luckily, apart from RRMNF, we had a whole bunch of derivatives and isomers based on the same scaffold. And when you look at this carbon atom, it's actually quite special because it has 4 different moieties attached to it, so it's a chiral carbon. And you can have the same moieties attached to it in a different orientation and get a different compound. So when you rotate the 2 moieties, you get RS MNF. And we looked at the downstream signaling, downstream out of beta-2 adrenergic receptor, looking for the ability to activate G proteins and beta-arrestin. And what we got is that these compounds are actually quite different in the way they act on the receptors. So both of them are still antagonists of GPR55, but they activate beta-2AR in a different manner. So RRMNF is like a classical agonist. It binds to the receptor, it activates it, and it triggers all signaling cascades downstream of this receptor. But RS, it's also an antagonist. It can bind to the receptor, but it specifically activates only one pathway over 2 other pathways. So it's a biased ligand. It displays functional selectivity. So we tested it as well in some simple assays, looking at the expression in microarray experiments. We observed that this compound can suppress the expression of several biomarkers, cancer biomarkers, including hyposignaling pathway and Wnt beta-catenin. And we were able to confirm it by Western blotting. And then we carried out another xenograft study using PANC1 cells. So this is the growth of tumors in vehicle-treated animals. And RS-MNF effectively and dose-dependently suppressed the growth of this PANC1 tumors. And we believe that it can be used as a support compound for classical cytotoxic drugs like doxorubicin. So to sum up, again, you should never be overexcited about some initial results, but it's good to persevere and continue the research. Another thing is that small differences in structure, like chirality, can translate into large differences in functional outcomes. And it is possible to create drugs that will not only target a particular receptor, but also a particular signaling pathway downstream of a receptor. And trying to get a link with previous talks, from our preliminary data in syngeneic cancer models in mice, we learned that inhibition of GPR55 can have some immunomodulatory actions, as we observed increase in cytotoxic T lymphocyte count and drop in the number of M2 macrophages in animals treated with RS-MNF with inhibitor of GPR55. So maybe this can be a new target for better immuno-oncology. So closing my presentation, I would like to thank my mentors and colleagues from Medical University of Lublin, NIH, and other universities and institutes. Thank you.
Thank you. And questions? Professor Nowis?
Okay, so in general, the project initiated to get something that will help with congestive heart failure. And that was the initial idea of developing selective beta-2 adrenergic receptor agonists. But again, we did some preliminary experiments in different types of cancer cells, and we discovered that when you increase the amount of cyclic AMP, which is the classical effect of beta-2 adrenergic receptor activation, you get an inhibition in growth of the cells. So we followed that. So it was never the main idea to get, let's say, anti-cancer agent.
Okay. Dr. Stoklosa?
Honestly, no, this was never studied. So the pathways that we were recently focused and which we get some positive data is HIPPO signaling and the APTAs. So, but maybe we should focus on this and at least check. Okay, thank you.
Okay, thank you. More questions? If no, I would like to end this session. Thank you for the nice presentation, for discussion, and we are now to have coffee break. Thank you.